Wonderful. Thank you very much. So thank you, Shura. Thank you for the kind introduction. And thank you everyone for coming here today. I'm very happy to be here and have the opportunity to present you research from my lab here at the Center for Soft Matter Research at NYU. So as my title slide already suggests, the next hour or so, we will be diving deep into nuclear physics. But it will be a different kind of nuclear physics. It will be the other one, meaning the nucleus at hand won't be atomic, but cell nucleus. And this is a topic that has been fascinating physicists for a very long time. Already, I mean, many of you are familiar with a book by Erwin Schrödinger, What is Life, where he has pondered upon how can we actually describe the events in living cells by physical laws. And he proposed that the fact that if we cannot immediately explain these events in living organisms by physical laws currently existing, then the reason might be not that these events defy these laws, but the physical laws that describe them have yet to be discovered or revealed. More specifically, physicists have been also very much excited by genome for many, many years. In fact, seminal work by Rosalind Franklin, James Watson, and Francis Crick gave us the structure of DNA double helix. And it was a physicist, Francis Crick, who also postulated the central dogma of molecular biology that suggests that there is a universal flow of information in all living organisms. What it means is that there is this universal flow of information from DNA through RNA to protein, where the genetic information is encoded always in DNA molecule, then it's copied or translated into RNA intermediate, and then further translated into protein, which is the molecular machinery that then carries out biological function. So let's focus here in this talk on DNA. In human cells, we have actually about 2 meters of DNA inside of a cell nucleus that is barely 10 micrometers in diameter. So you can imagine that was a true engineering task that nature needed to solve. How do you pack such a gigantic length of a molecule in structure so small? Here, I let you ponder for a minute, consider that you have 2 meters of DNA in every single cell of your body, right? So this is an enormous amount. So how do we pack that? Well, nature has solved this problem by packing the DNA molecule around protein particles, around proteins histones, forming here this pearls. So this structure, you see this pearl necklace, you have these beads on a string structure, and this pearl necklace is referred to as chromatin fiber. So you can think of it as a functional form of DNA inside of a cell. So now the question is, how many of such fibers would we have in cell nucleus? So here in my purple nucleus, you see here this yellow chromatin fibers. Well, one chromatin fiber corresponds to one linear DNA molecule, actually represents one chromosome. Now we know that in human cells, we have 23 pair of chromosomes, or 46 chromosomes. So we have 46 chromatin polymers present. Now in thermodynamic equilibrium, if you had a solution of 46 polymers, what would you expect? How would they be arranged? So for that, here I call for the image of mixed colorful spaghetti that might illustrate this situation of a polymer mixture in thermodynamic equilibrium, where you see that these spaghettis are totally mixed, obviously maximizing their entropy. Well, this is what you would expect inside of the nucleus, but that's not what you find. How your chromosomes are actually organized, they sit very neatly packed, every one of them in their little region, in their little corner, or as we call it, chromosome territory. So this is the first sign where you see clearly that this system is actually out of the equilibrium. So as I already alluded to, genome is actually organized. So your chromosomes are sitting in these well-defined chromosome territories that has been revealed within past 20 plus years by fluorescent in situ hybridization, which is a sequence-specific staining of genome. Moreover, over past 20 years or so of techniques called chromosome conformation capture techniques have revealed defined spatial interactions in the genome. This is a biochemical assay that measures proximities or probabilities of proximities of specific sequences in space. So that allows us to render actually 3D structure of the genome, and you see our present picture of the folded genome is actually greatly detailed. In fact, the resolution that we presently have is about 1 kilobase pair. That is amazing. However, these both pictures that I'm showing you here are static, meaning they correspond to one snapshot in a life of a cell. So the question that appears is, how do you reconcile it with this? So what you're looking here at is a human cell nucleus where the genome is now fluorescently labeled green. So all of this here is DNA. And as you can see, this is a live nucleus. There's a lot of motion. There's a lot of jiggling. There is a lot of flows and so on. And this, again, this is going on right now in every single cell of your body. So there is a lot of dynamics, a lot of motion. So what we are trying to understand in my lab are actually the physical principles underlying the organization of the genome and dynamics of the genome in space and time in living cells, which is a beautiful non-equilibrium system. So specifically, I will divide my talk into 2 parts. In the first part, I will show you what have we learned about flows and dynamics of the genome over the past few years in my lab. And then in the second part, I will show you how the knowledge, what we have obtained about the dynamics, how we can actually use it to extract material properties and rheology of the genome, as well as other nuclear components. As here, the example shown here is nucleolus, a structure that I will introduce later. Okay, so let's start with part 1, genome flows and dynamics. So when I entered this field, the approach to measuring genome dynamics was to fluorescently label a protein of interest and follow it with a single particle tracking as a function of time. Now, this obviously is highly informative, but it limits you to how many dots, how many proteins actually you can track in time. So what I wanted to do was to actually map chromatin dynamics simultaneously across the whole nucleus in real time in living cells. So how do you do that? So for that, when I was a postdoc, I developed displacement correlation spectroscopy, a method that allows you to do exactly that. It uses fluorescent labels that I put on those pearls that I introduced you in the structure earlier. And what I'm doing here is basically I'm monitoring intensity fluctuations of these fluorescent labels in time and in space, and in a temporal spectroscopy-like fashion that allows me to determine actually maps of motion across the entire nucleus at different time scales. So here, for example, 2 examples that I'm showing is the short time scale and long time scale of such chromatin displacements. These vectors here are color-coded by direction to kind of show you how are these motions correlated or not. And you see at short times, the motions are clearly uncorrelated, they look random. However, when you go to long times here of several seconds, you start to clearly see regions of same color. So basically large chunks of chromatin that actually move together coherently across the nucleus. So here, kind of an outline of like a little cartoon. If I would stop this movie here at one snapshot, you would see such a map at a given time. We can, of course, measure the size of these regions by calculating the spatial autocorrelation function. And we find that genome exhibits coherent motion on micrometer scale. Now, I have introduced you this picture before of our chromosome territories, which when you look at my map of dynamic coherent regions, looks extremely similar. So of course, the first question that occurs is, are we actually looking at the motion of chromosome territories as such? So I performed another experiment where I have, in addition now to my entire genome, I have now also obtained chromosome territory staining. And now I can obtain basically their contours, their outline, the boundaries between territories, as well as my maps. And what I find is that here you see in this cartoon, you find that what occurs is that you would have two territories, so two polymers, the purple and green polymer that are touching here, and the coherent region, so the red arrows, they actually span across the boundary of the two polymers. So to put it into big picture, what you see here, when you have these two neighboring polymers, here the purple and green, the coherent region actually spans across them, right? And so the whole nucleus is kind of covered with these type of coherent region. They live for several seconds and they break up, new ones form, and so on. So, but to understand this, right, that was extremely tantalizing discovery, and I have been actually ever since on a quest to understand it and reveal where does it come from. So the first questions that we've been asking is this a property of active systems, or is it something that also a passive system could undergo? So first, to address this, what we can do, we can indeed observe our system in its passive and active state. Active state would be when all the natural processes are happening and I'm in a living cell. Passive, I can achieve passive state by actually depleting ATP, so the fuel of my active processes in the cell. And that means all active processes cease and I can observe just thermal fluctuation in the system. We can, from the DCS maps, from those displacements, we can obtain a mean square network displacement, which basically tells you the mean square displacements as a function of time. And you see here, this calculated for active and passive case, there is clearly a big difference in the dynamics. The dynamics is strongly reduced once you deplete activity and the system remains passive. We also find that the active behavior is subdiffusive. So for those of you interested within the subdiffusion exponent of about 0.7. So moreover, from our maps, we have the information, right, about our displacements, also their spatial localization. So what we can do, we can also compute power spectral density and look specifically at which wavelengths does the activity contribute. And what we find here, so in this plot, you see different colors correspond to different time scales. The solid lines show the active cell, the active nucleus, the dashed ones is the passive case. And you see here at large wavelengths, there's a huge difference upon ATP depletion. So you see that the activity here strongly contributes actually at large wavelengths, which is exactly right, the micron scale coherence that we see. Our regions of coherence are 3 to 5 microns large. So we started to ask, where can this coherence actually, where can this come from? And what we find, the way how we can think about it, we can classify the types of events that can occur into two types. First, imagine you would have a vector event. So basically that would be an action of an enzyme that could be described by a force dipole. So there is a force that has a direction as well as magnitude. And specifically, right, it will have here this shape. So here I'm mentioning some examples of molecules that actually could perform such function. The other type of active events could be scalar events, where you have the concentration fluctuations of basically condensation and decondensation of the polymer that you see across the nucleus. And also there are chromatin remodelers, molecules that could do that. So we have built this framework together in collaboration with Shura Grosberg, Yitzhak Rabin, and Robin Brunsma. And based on our experimental data, we've developed a hydrodynamics-based theory that describes actually the active chromatin dynamics with these two types of events. So just briefly, this theory accounts for chromatin and nucleoplasm in terms of a two-fluid model, with chromatin being the polymer, nucleoplasm being the solvent, and then for the activity in the form of these two types of events that I have just mentioned, so the vector events and scalar events. And what I want to here stress is the most intriguing result, and that is the prediction that it's the activity specifically in form of force dipole dipoles that can lead to large-scale chromatin flows. So this is what is here happening. You see when these force dipoles start to hydrodynamically interact, they can actually lead to these large-scale actual alignments and large-scale chromatin flows. So this was very exciting and intriguing to us, because that gave us kind of a mechanistic picture of what are the molecular players, what is the source of the coherent motion that we're looking for. So we started to explore it even more, and together in collaboration with Mike Shelley and David Santillan, we have built a model of interface chromatin that now models the action of such force dipoles. So how do we do that? So in this model, we model the chromatin as a chain that is confined within a spherical nucleus. This chain undergoes thermal fluctuations, but in addition, we apply these force dipoles at all these red patches here that you see. And so we observe the dynamics of this chain with these dipoles acting on it. Now, this approach allows us to study separately the effect of contractile and extensile dipole. And you see their effect is very contrasting, right? Here, in contrast to Brownian chain, you see that contractile dipoles kind of behave in Brownian-like way, but look accelerated. But extensile dipoles are very different. You see how they actually extend the chain. Now, when you look in the confined case here down in the bottom row, you see for the extensile case that indeed the extensile activity leads to these large scale alignments, these arrangements. And here, these movies, obviously, they show you the motion of the chromatin fiber, but what we actually compute in the numerical simulations are also the flows of the fluid, right, within which the chromatin fiber is embedded, and that actually mediates the hydrodynamic interactions. So let's compare these results with our experiments. So what we see in experiments is, right, so that in our passive system, actually there is no coherency presence. So you see here uncorrelated motion. In our active system, the chromatin does move here coherently. This can be here very simply and beautifully visualized by the autocorrelation function, the spatial autocorrelation function here for the passive system. That's the blue curves. You see they're all here collapsed for different time scales. At the bottom, there's no correlation. But for the active situation, the red curves show actually a strong coherency here present. So let's look at our model. Now you see clearly that in the passive case, again, already from the DCS maps, there is no coherency present, and you clearly by eye already can see large coherent regions, so patches of the same color for the extensile dipoles. Again, we quantify it by the spatial autocorrelation function, and what we find indeed, right, for the passive case, no correlation. For the extensile case, we find a large scale coherency that occurs. Now for the contractile case, note that those are the green curves here in between. There's a very small correlation present, and that actually corresponds directly to the size of the Stokes slit, right? So of the local flow caused by a single dipole. So what basically this model shows is that hydrodynamic interactions between specifically extensile dipolar forces can lead to large-scale coherent motions observed in vivo. So where that brought us, right? So now we had this framework of force dipoles, and we knew that it's the extensile activity that is needed. So how can we test actually a contribution of a single active site? How can we do that actually in living cells? So we decided to do that by looking at a single gene and its activity, and we wanted to toggle it on and off and observe how does it change and how does it contribute to the motion of the surrounding genome. Okay, so how do we do that? So to visualize a single gene in vivo is no simple feat. For that, Fangyi Cho, a graduate student in my lab who graduated at the beginning of this year and has started recently her postdoc at UPenn, She has built up this beautiful system implementing CRISPR technology that allows us to do that, exactly that, to visualize single genes. Specifically, so here, for those of you interested, we actually went wild and went for both repetitive and non-repetitive sequences. So for those of you CRISPR enthusiasts, you might like the detail. But here in big picture, so we chose to visualize 3 different entities, 2 genes, IL-6 and MUC4, and telomeres. Those are physical ends, free ends of our chromosomes. So those are kind of nice punctate structures also as a control for us to look at. And you see here, we successfully visualize here these dots here, right? So this is the successful here CRISPR signal that we then verify through other types of staining that indeed it works and it stains the genes, the sequence specifically that we're after. And here, as I said, in telomere case, these dots here that you see, those are free ends of your linear polymers. Okay, so once we visualize those, then we can perform our dynamic experiment. So on our microscope, we have built a custom-made beam splitter where we actually enable our microscope to see in 2 colors at the same time. So it has kind of a double vision in 2 different colors. And we built these high resolution images in real time simultaneously. We obtain 1 signal from chromatin, so from the stained fluorescently labeled genome. And the second signal here comes from CRISPR. You see here this dot, that's the gene of interest. And now the analysis that we perform here, we can follow the motion of the genome via DCS around it, and here confirm with correlation functions that indeed the coherent motions for the genome-wide motions is here. And the gene we are following here with single particle tracking, and we're looking at its trajectory. And what we can do here is we can simultaneously monitor the motion of a single gene in the genome while measuring and visualizing also the flow of the nearby genome. So this is truly amazing. This is actually happening in living cell. So the cell is alive and happy, and you can bring it back to incubator and look at it the next day, and it's still alive and happy. So you get here a healthy physiological behavior of how our genes here behave. And so we can learn and extract here beautiful physics out of it. So as I told you before, the genes we chose in a way that we can actually toggle their activity into an on and off state. So those genes specifically that I have shown you, we know how we can actually enable their transcriptional activation, so we can make them to be on. So we then observe their dynamics in both inactive state and active state, and look within the context of genome how that motion changes. Moreover, for example, how does the correlation of the motion of the gene versus the surrounding genome, how does it change? And indeed, what we find is what we see is that the motion on active genes is strongly correlated actually with the flows of nearby genome, where when your gene is inactive, such correlations are not present. So it's actually, it does not move together with the surrounding flows. So this is actually consistent with our picture that the activity at the gene level indeed drives genome-wide motions. So far, what I have shown you here in this part was the motion of a healthy genome and of healthy genes. Now, what is happening actually frequently, however, in our genome is that it experiences DNA damage. That happens normally in biological processes, but also due to environmental factors, as from radiation to the UV light, as when you walk on sun. These type of damage and breaks, for example, here specifically, I illustrate this would be a DNA double-stranded break, we call it, they can occur. So what we have been asking ourselves, how does such DNA damage, these local perturbations, how does that actually affect chromatin dynamics. So this is a beautiful work by Jonah Eaton in my lab, who has very carefully introduced this type of DNA damage of these double-stranded DNA breaks into human cells. And specifically, we use a marker that allows us to visualize that the damage. So you see Jonah can titrate, so control the number of these damaged sites. So each of those red dots corresponds actually to a cut in your DNA molecule. So you see here, as the number here increases, we observe the dynamics. So what Jonah here has done, he follows the motion of these double-stranded breaks, and that's not really simple, as the signal, of course, you see they vary in shape, size, intensity, and so on. So Jonah has developed this beautiful machine learning assisted algorithms that actually really allow us to detect and track in real time all these the DNA damage sites in live cells again. This algorithm has enabled us actually to obtain the largest study to date. We have analyzed over 16,000 damage sites in over 600 nuclei. And so what we have learned from analyzing these type of trajectories of these DNA damage sites is that they move actively, they move subdiffusively, and their motion is very unique and very different from a chromatin that moves in a physiological way, so in a healthy way around them. We find their mobility is much higher of this damaged chromatin, and to a certain extent, actually the constraint is higher. So in terms of subdiffusion coefficient exponent, that one goes down. Moreover, we find that actually the polymer, so the genome, assumes a very unique type of packing at that DNA damage site that leads to this unique compaction that also allows you actually beautifully identify which sites of genome are actually experiencing damage when you observe it. So this is extremely interesting, as it gives us the whole dynamic picture of the active physiological processes as well as the damaged one. So with that here, I'd like to just briefly summarize what I have told you so far in the first part of my talk, that I like to view the genome kind of based on its active behavior as being self-stirred. You see, it's hugely dynamic. There's a lot of active processes that are responsible for that. These motions are subdiffusive, and there is coherency. We found that this coherency is an emergent property of activity. So passive systems do not exhibit this type of coherency. Moreover, we find that it's the hydrodynamic interaction of extensile dipolar forces that cause that. And the activity that leads that coherency is actually activity at the gene level. So you can imagine that this type of mechanistic picture, right, has huge implications for genome organization and gene regulation as such, because that suggests, right, imagine all these dynamic motions that the genome undergoes that will change the organization of the genome in space and time. And in terms of the gene regulation, meaning also the information flow, which genes will be accessible for processing and which not, that will be highly impacted with the spatiotemporal dynamics of the genome. So with that, I will switch gears a little bit to nuclear rheology, but not so much actually. So I have told you now a lot how we study dynamics and motions inside of human nuclei in live cells. But now I will show you how can we actually use these dynamics and extract material properties and rheology of genome and other nuclear components. Okay, so if we want to obtain material properties of the nucleus, so how can we do that? So for all physicists here in the audience, everyone's familiar. If you want to study this type of nucleus, what do? You bombard it with other particles. Well, what do you do with cell nucleus? Something very similar. You bombard it with particles. But instead, now what we have to do, we have to inject these particles, get them inside, and then observe, right, the interaction of the particles inside with the nuclear interior. Now, this, as you can see, so in my high-tech animation, the needle was actually drawn to scale, meaning the needle is really gigantic. It is basically, you know, couple microns. The particle is at the order of, you know, half micron. And remember, the whole nucleus was 10 microns only. So meaning if I inject this particle with this type of needle, well, the wound that I leave will be dramatic, suggesting, right, that this is a rather invasive method. So this has been done in past, and we actually, in fact, we've done it in my lab as well. We've injected ourselves, but we saw that this is a hugely invasive approach. So how can we do that in a non-invasive way? So what I was thinking, well, have a look. There are already probes already present inside of the nucleus. So why don't we try to use those? So you see here, this big spheres here. I will tell you in a minute what it is. So here specifically the nucleolus, there is a probe present, and we will be able to use it for rheological measurements. So what we have developed then here from now on, we develop a novel strategy how to actually measure material properties and rheology of the nucleus and its components by using actually intrinsic dynamics of its components. So I will show you how can we study intrinsic dynamics of the genome and other nuclear structures and measure their material properties from that. Okay, so the first candidate that I mentioned already is nucleolus. Why did we focus on nucleolus? Because it's the largest structure present, right? So you could see it right away there in, even when it was not specifically stained. Here we use a fluorescent dye that visualizes nucleoli. Nucleolus is a membraneless structure that makes ribosomes in cell, and ribosomes make proteins. So it's very important organelle inside of a cell. So you see these guys here, they appear large, but think again, the whole nucleus is 10 microns. So the nucleolus is generally 1-2 microns in size. So it's actually teeny tiny. So we observe them very closely with high spatial and temporal resolution. And what we have found was that its shape actually fluctuates very subtly over time. So it does change shape. And this was here work by a former undergraduate in my lab, Shannon Haley, who's now a graduate student at UC Berkeley, who has written this beautiful code that has allowed us very carefully extract these surface fluctuations from which we can actually compute here. Well, we can visualize those. So here I'm plotting them as u square, right, as a function here as a polar angle. So what you see here, those are fluctuations at different time points. The different colors are different times. So it's really, it has these little kicks on the surface. And if we assume that these kicks, for example, would be thermally driven, we can generate an estimate of a surface tension of the nucleolar surface, right? So of the interface between the nucleolus and the surrounding chromatin, the surrounding genome. And what we find, the surface tension is at the order of 10 to minus 6 Newton per meter, which is extremely low. It's several orders of magnitude lower than water-air interface of your water droplets in air. And that means that it could not be by any means, for example, solid particle, but this is a very much consistent with a liquid droplet. That, of course, makes very much sense, as this structure is membraneless, so it lacks any boundary that could actually hold it together. So we see that this behaves as a liquid droplet. So another dynamic event that we decided to focus on is to look actually at the fusion of these nucleoli. And since we know now that they behave as liquid droplets, specifically, right, at their coalescence. So this sounds simpler than it is, because this is a truly a big endeavor, as the number of nucleoli of this size per nucleus is very low. You generally have, as you can see here in this picture, 2 or 3. Then they're tethered to DNA, meaning they don't move actually very much. And they fuse once per cell cycle. Now, the fusion takes about 10 minutes, and the cell cycle is at the order of 35 hours. So you have to be kind of at the right place at the right time, not to miss it, and to observe it. So here, fantastic experiments have been performed by Christina Karagin, a very talented grad student in my lab, who actually graduated a couple of months ago and now started her postdoc at the New York Genome Center. So she has developed this semi-automated algorithm or approach where she actually scanned through 10,000 cells, where she identified 150 fusion events. So those were nucleoli in a very close proximity, which were very likely to come together soon and fuse. And 14 of those we were able to capture actually in XY plane. Why do we care about XY plane? It's because that's where we have the highest spatial resolution, right? So because of the way how the point spread function is shaped, we basically our Z resolution is lesser than the XY resolution. So that's why we focused here on those. And so let me show you here how such experiment looks. So first of all, I want to note that this is truly a coalescence of smallest droplets ever seen on this earth. So you can see this is how such experiments look. We observe the nucleus. You see here these two droplets coming together. And what we are measuring here is this neck connecting the coalescing nucleoli, right? So you see here it's this neck in between them and you look at its growth. So let me show you here. Those are these 14 events that Christina has recorded. That are extremely informative. Why are they informative? It's because the kinetics of the growth of this neck provides exactly the information on which forces are actually participating in the coalescence event. So since we know in our system here, we are at very low Reynolds numbers, meaning it's the viscous forces that are dominating the coalescence, there are still two possibilities, viscous forces of which fluid can be dominating. If an outer fluid, so if the outer fluid of the nucleoli would dominate, so in this case, the nucleoplasm or the chromatin solution around it, then it can be shown through theories that the growth of the neck grows as t to 1/2, so with this specific power law. In contrast, if it would be the inner fluid, so the nucleolar fluid, whose viscosity dominates, then the coalescence, the growth of the neck, would grow linearly. So we can rescale our data by the size of our nucleoli and also to the exact same starting time. And what you see, our data beautifully collapses on a power law of t to 1/2, showing us that indeed it's actually the outer fluid that controls the kinetics of the nucleolar coalescence. So the situation that you have here, what you're observing is basically having two water droplets immersed in honey. So you can imagine it will take them for a while, right, to coalesce and to fuse. They're slowed down. But if you take those same water droplets and let them actually coalesce in air, they will coalesce very quickly, right? Very fast. So this is here, this honey water situation. So you see it's the chromatin dominating the kinetics of nuclear coalescence. Okay, so this is actually very powerful piece of information because it allows us further actually to extract the ratio of the surface tension of the nuclear interface, nuclear chromatin interface, and the nucleoplasmic viscosity, so the viscosity of the surrounding fluid. So we can do that, we can extract it for all of our 14 events. And don't forget, we have previously already obtained in a separate measurement actually the surface tension of our nucleoli, which allows us now to actually determine the viscosity of the nucleoplasm directly. So here what you can see is that we can use nuclear coalescence as a completely non-invasive rheological probe of chromatin. So by extracting the physics out of this dynamic behavior, we can actually obtain material properties and its biological behavior. What I want to stress that this is really totally non-invasive, meaning we have only looked at those cells. They did not even know we were there. And everything what we have obtained is basically from a pure observation of these dynamic events by, of course, visualizing the right entities. So this strategy of using intrinsic dynamics to extract rheology, we have done actually several times for different nuclear components quite successfully. We have looked specifically here, Fungi, Chu, and Shannon Haley in the lab, they have looked at the fluctuations of nuclear envelope. So when you can see when you look closely here at this edge, at the rim of this nucleus, you see how its surface here beautifully undulates. So we can analyze these fluctuations and understand them. We also find they're actually actively driven. We determine the thermally driven part and actively driven part, and from those, actually from the passive part, we can determine its bending rigidity, the bending rigidity of the nuclear envelope. In fact, we find that the rigidity increases as the cell ages. And we also actually were able to determine here the molecular mechanism. So what are actually the forces, which forces are driving these fluctuations? In another project, Alex Clavijo in my lab is looking at motions of the polymer free ends, the telomeres, and also of at the geometric centers of our polymer, which are called centromeres, as illustrated here in this cartoon. So the blue guys and the red guys, and you can see, so he can visualize those again simultaneously also with the motion of genome, and he can extract basically rheology across the nucleus from the motion of this structure. Moreover, here Nikitas Kanalakopoulos, MD PhD student here at NYU, and Stephen Ionov, a former undergrad in my lab, currently a grad student at Dartmouth, they have visualized actually several more additional structures inside of the nucleus, where we have focused on understanding the heterogeneity and organization in the context of dynamics. So it's truly the picture of dynamic organization that we are here trying to make physical sense of. Specifically, we have focused actually on the heterogeneity also of the genome, so of the polymer itself. As you can see here from this yellow cartoon, from this yellow line here, that kind of is supposed to illustrate how the genome, how the polymers are heterogeneously distributed throughout the nucleus. You see that my green signal is very heterogeneous. So you see the solution is not homogeneous, but heterogeneous. And this is something very important that I will be speaking in a minute about, and specifically a system that allows us to study the heterogeneity of the genome and to understand that it's actually a genome during cell differentiation. So for that, first, let me remind you, so what is cell differentiation? That is a process by which stem cells become specialized cells, right? So the stem cell becomes a neuron and becomes a blood cell, or so on. So what is very interesting about this process is that during cell differentiation, a cell nucleus and genome inside undergoes a dramatic reorganization. What it means here, as shown in this cartoon, in stem cell nucleus, the genome is actually quite homogeneous, beautifully here just spread out through nucleus. Once the cell differentiates, in the differentiated nucleus, the genome starts to be heterogeneous. And you see here the structure of higher density, of higher compaction occurs. We refer to those, we call them heterochromatin. So we have started to study this type of system in my lab, and specifically this is a project led by two graduate students, Irash Eshgi and Jonah Eaton, where they we use actually mouse embryonic stem cells with, again, fluorescently labeled the genome, where we observe the dynamics in those mouse embryonic stem cells. We then differentiate these cells. So we let them go through the process of differentiation and look at them again once they're differentiated. And here specifically, we are differentiating them into neuronal cells. So what you see here, it's then the differentiated version of our system. So you see the difference is striking. The size of the nucleus is completely different, and the distribution of the genome inside is very different. It might be difficult here to view from this signal on a first look. So let me color it differently. So we have, from the way how we are staining the genome, we can basically, from the intensity of our fluorophores, we can directly obtain and measure the compaction, right, or the density. So you can see, if we now observe these densities here, you see for in the stem cells, it's mostly the genome is mostly in the red region, so relatively low compaction states. But you can see clearly an appearance here of a new yellow phase upon differentiation, which has quite high, a much higher compaction. We can quantify it beautifully, right, over basically many cells here. Those are histograms of chromatin compaction over a population of undifferentiated nuclei and a population of differentiated nuclei. And so you can look at this as a kind of distribution of density states. And you see that this distribution upon differentiation has strongly shifted to the right, and that there is a new phase present, right? So these states here, these density states that are only present in the differentiated nuclei, but are basically as good as non-present at all in the stem cells chromatin. So we see that we have upon differentiation this new high compaction phase that emerges. So we have moved further and started to analyze this phase also dynamically. For that, we use our displacement correlation spectroscopy and again obtain dynamical maps of the genome in both undifferentiated and differentiated states. And you can imagine this is extremely rich in its information, right? So there's a huge data richness that is in these maps. So I will show you only some of the indicators that we have read out. So for example, you can look at local speed. You can here compute, this is an average speed over a certain time interval, an average speed that is basically at certain site experience. And what you can see here right away is that while the stem cell is mostly bluish, so has here this higher levels of velocities, you have this appearance of yellow slow dots here inside of a differentiated nucleus. We can do even better. We can look actually at local correlation of the displacements of the chromatin. So how do we do that? We look basically at every single vector here inside of our DCS map and calculate the spatial correlation with its neighbors and measure basically the exponential decay with the distance. How fast does it decay, right? The correlation with the neighboring vectors. So you can obtain such a correlation length here again for each measurement site, and you see again that you start to here in the sea of pink, which is a low correlation state, you start to observe here patches of yellow, which are high correlation. So what you can see already on a first look, that it appears as if there was nice correlation in terms of that the highly correlated states are actually the slower one. And don't forget, we can also actually compare it with the compaction that we have measured before. And indeed, just by visual inspection, you already see that these yellow regions across all of these three maps are actually in very good agreement. We could, of course, quantify it and look at it as a speed versus compaction and correlation versus compaction. And indeed, what we find is that there is this second phase that is present upon differentiation that is highly compact. The genome is less mobile inside of that phase, and displacements of that phase are highly correlated. This is consistent with a solid-like gel phase. In contrast, the surrounding phase around has much lower compaction. It has high mobility, and the motions are uncorrelated. So this is consistent with a liquid-like sol phase. So what we are seeing here, though, which is truly amazing, is that really genome is compartmentalized via rheological phase separations into sol and gel phases. Now, we can go actually even further, right? So now that we have determined that we do have two rheological phases, we can actually test even the bulk behavior of our two types of chromatin, and we can do it in the following way. We can analyze the chromatin dynamics in both undifferentiated and differentiated case the same way as I showed you earlier in my talk, by computing the mean square network displacements. And you see that already here, the dynamics appears very different between these two states. The stem cell chromatin, the undifferentiated one, has larger displacements, right, over the same time. So it's faster, it's more mobile. So what we can do, we can use the concepts from passive microbiology, and using generalized Stokes-Einstein equation, we can actually translate these mean square network displacement into the viscoelastic modulus and its storage and loss moduli parts. Now, of course, this is a passive concept, and I have just explained to you how active the system is. So we have to hear what we're doing in order to be able to use this concept. We are using an effective temperature to account for this activity, right? So that means that we are not extracting an absolute value of our G prime, G double prime, but a functional shape of it. So what you can see here right away is that this is when you just compare the two colors, the red and blue curves, the solid ones being the elastic moduli, the dashed ones the loss moduli. So what you see clearly, they're very different for differentiated and differentiated chromatin. Could that difference be indeed coming from the fact that, as I just showed you, that in undifferentiated cells, you have only one phase, the sol phase, and upon differentiation, you have emergence of a second phase, the gel phase. So actually you have a biphasic material, which has a sol and gel phase. Can we show that? So for that, we started to model chromatin rheology. And first we looked actually even closer at our rheology data. And what you see when you compute the loss tangent, which is here this tangent delta, that is the ratio of your loss modulus to your storage modulus, we see this beautiful power law here of omega to -1. Now, this is this was very exciting to us, because this is indeed a scaling that is found for Maxwell fluid, which is a simple viscoelastic fluid with one single relaxation time. So that was actually very exciting, and we fitted this model immediately here. As you see, the simple viscoelastic fluid can be mechanically simply represented by a dashpot and a spring in a series. And so when you fit it, you see, I mean, it's difficult even to see the fits properly because they agree with the data so well. So you see the lines here on the top of our data are beautifully describing actually the undifferentiated chromatin as Maxwell fluid. So what happens now upon differentiation? So here clearly, so note this dashed line, that's the Maxwell fluid scaling here in this case for the loss tangent. Clearly it's not Maxwell fluid, it's very different. So what is it? So what we were looking for was a minimal model that can account for the emergence of that solid phase. So what we started to do, we have started to add to the Maxwell fluid a very simple solid. And this is here, in this case, a simplest solid is a Kelvin solid. And a material where you have a Maxwell fluid and a Kelvin solid here in a series, as you see here in this cartoon, is called Berger's material. So then we fit the viscoelastic modulus of this material to our data, and voila, you see we have a beautiful agreement here with actually two phases with the sol and gel phases here in the system. So what we found here, that indeed undifferentiated chromatin can be described as a sol phase, as a liquid-like phase, and upon differentiation we see that the genome can be described by a coexistence of two phases, sol and gel. And there is the gel phase, in fact, appears locally via sol gel phase transition. So we see, right, originally from a purely liquid-like chromatin, now you locally form these gel patches. So this is really striking because it shows you how dramatically actually material properties of the genome change, right? So we see that the formation of this heterochromatin is really directly connecting actually with the formation of a new physical phase. So this brings me to the end of my second part, which I will just briefly summarize here with respect to time. So what I hope I was able to show you, so we have developed this novel strategy how you can extract rheology from intrinsic dynamics of nuclear components that allows us to actually obtain material properties of structures that we had not even dreamt of, right? I mean, there is no chance of measuring those otherwise, and we obtain viscosity, surface tension, moduli, and so of different components. We have also found that the genome is compartmentalized via rheological phase separation. Moreover, we found that it's a local rheological phase transition of the genome during cell differentiation that leads to formation of the new phase. And those phases are, right, the sol phase before differentiation and the sol and gel phase after. So this is truly this finding has a huge impact on our understanding of genome dynamics and the genome organization and compartmentalization, as you see that it's the nuclear material properties that are changing. Those, when you remember back to the central dogma that I mentioned at the very beginning, the nuclear material properties actually through the central dogma impact all cellular processes. What do I mean by that? Every cellular process must start by central dogma, right, with DNA. So if you actually change the time scales and length scales of the genomic interactions, here, for example, by forming a rheological phase, then you're actually impacting largely all cellular processes. So with that, I would like to give big thanks to my team. It takes a village. That was a huge amount of work done by a beautiful group. I have been very fortunate to nucleate over the past 6 years a dynamic and vibrant group of chromatin enthusiasts. So you see, we've been having a lot of fun. This is how our life used to look like. This is how it looks these days, which I guess we are all familiar with. So in addition to all current and past members of my lab, I would like to thank our collaborators, our funding sources for supporting us in these endeavors, and you for your attention. Thank you, and I'm happy to answer any questions you guys might have.
Thank you very much.
Thank you, Alexandra. Any questions?
Can I ask a question? One very simple one. Does it matter what kind of nuclei? I mean, does it matter which kind of cell is it? I mean, since there are so many, or...
So that's a fantastic question that we have been asking ourselves as well. And it's amazing that there are certain general universal principles that actually translate across. And this is actually one of our other projects that we also have, that we have looked at a panel of different genomes over different cells. And we're actually trying to understand the general physical principles underlying there. And I don't want to here reveal too much yet, but it's looking very promising that actually there's a universal physics behind all of it. So while the scaling will change, by understanding the properties, the material properties of the specific genome, you can actually find the same underlying principles.
Thank you.
Any other questions, please? There are.
Sure, I raised my hand. Should I just go?
Yes, go ahead, please.
Okay, lovely talk, Alexandra. So back when you were talking about the mouse embryonic stem cells,
Yes.
one thing that struck me was that the nucleus of the stem cell seemed to be just have so much smaller volume than the nucleus of the differentiated cell, and yet it has to have the same genome inside it. Is the difference that the differentiated cell is just full of protein? What's the difference?
That is a great point. You have answered your question already. Indeed, basically, upon differentiation, you're building all these other additional structures, right? You are organizing the genome in a very different way. The stem cell is a kind of minimalistic, very simple system that has to be very fast and very accessible. So you keep kind of minimum of stuff present, right? And basically, that's also why the genome is in this open, low density state, because all genes have to be equally likely to be able to be accessed and expressed and used. Where once you differentiate, you kind of pack away the genes that you don't need, kind of like a winter clothes into the heterochromatin, and that is packed away. And also other cellular structures are being formed, other nuclear structures that then take up place. So yes, so that's it's amazing how much larger actually the volume then becomes.
I see Michael Blanton and Glenys Farrer who have questions.
Yes. Michael Blanton is next question. Yes, I was.
Hi, thanks. That was amazing.
Yeah.
I had a question. Your second part, sort of expressed all the rheology in terms of sort of these kind of models, material models, but your first part was studying the flows in terms of like a very detailed, microscopic is the wrong word in this context, but like not a general model. And I'm just wondering, is there like a connection to be made there? Like, doesn't the sort of the model material that's appropriate, it must have to do with what's going on, in the material itself. But I don't know if that's an interesting question to ask.
Well, good that you're asking. So this is something actually that with Mike we're currently working on. So we are actually exploring for example, the heterogeneity now, specifically in the numerical model, in the simulations, actually then specific processes from what I have, so the heterogeneity that I have shown you, the formation of the heterochromatin, for example. So we can exactly use the model for that as well, and then compare how well does it describe it. So that's actually in works.
Cool. Mike Shelley, I guess.
Yes.
Okay, thank you. Glenys.
You kind of touched on the answer of what I wanted to ask, which was when it differentiates and you get these gel nodules, are they then identifiable as the active? I mean, it looked like just in the pictures that there were far fewer of them than there were genes, but maybe they were. Was there one-to-one correlation between genes and not?
Yeah. So think about it in the following way. So specifically in those gel patches, the genes, there are many genes packed inside. And what is in that gel specifically, those are genes that we call silenced. So those are the ones that are not being in use. So meaning if you are a red blood cells, you don't need neuron genes, right? So you pack them away. Okay. So you're completely right. There is many more genes in that patch, right? Also, what is very interesting, there is as I said, these genes inside are silenced, so meaning there is not an activity inside of those patches. So the gel patches are actually passive. Mm-hmm. So the activity is then actually around them.
Mm-hmm. Interesting. Okay, thanks.
Thank you.
It's wonderful.
All right. Anybody else, please?
How do you signal?
What? How do I what?
I just wanted to get recognized.
Oh.
So I can ask a question. In the first part of the talk, you talked a lot about the coherent motion. Is there a biological advantage that gives the cell?
So this is what has been my quest now for years, right? To get basically the physics as well as the role of it, right? What is it good for? So we don't know presently. However, because it needs to be present in every healthy cell, you have it. And since we know that it will lead to these large-scale flows, both of the polymer and of the fluid, I strongly believe that it will contribute, for example, to the distribution of molecular machinery, you know, inside of the nucleus that will be much more, that won't be diffusion limited, but you could actually add, for example, advection flows through these nucleoplasmic flows that will help you to move stuff, for example, through the nucleus. So it is possible that this might be one of the effects. It is also possible that, you know, it might allow during gene regulation, as you are changing these positions, if you have these various transcription factors and machineries that actually need to colloquialize kind of in the environment or in the neighborhood of several different parts of genome, that might allow them actually to get into contact faster. But those are pure speculation presently. Short answer, we don't know. Long answer, we have obviously several hypotheses and we would love to learn.
It sounds to me like short answer is we don't know, long answer is we still don't know.
We still don't know, but we love it, and we have we have a number of ideas that are being tested left and right.
Okay, left and right, very good. Other questions, please. I will ask my question. You said that this differentiation leads to sol-gel transition. I never know what is sol. And my feeling is that nobody actually knows, and this is a sort of obsolete terminology for the case when we don't know how to call this other phase. So the gel, we know it is percolated network of cross-links. And gel is a solid. Maybe it is soft solid, but it's basically solid.
Yes, yes.
And sol, I would still call it the liquid, but some...
Totally. So why I called it a sol, or I like to call it sol, right? So I'm using the Dejean terminology. So in his scaling book, so he speaks about sol, but you can also call it liquid. Call it liquid. I have called here, it's liquid-like phase. So we defined it here. Call it for now sol.
I am nearly speechless because I know Dejean's book pretty much in and out, and I don't think it's called sol.
I'll send you the pages that I'm referring to.
Okay.
What I meant, yes.
Okay. With a highlighter pen.
Yeah. Yes. Well, this is what we now do these days, all the time, back and forth.
All right, very good. Other questions, please.
One question. In the abstract, you mentioned ATP as the carrier of energy, etc. Now, there are cells that don't use ATP. I remember that even in some delicious animals like lamprey, right? That's so. But is there any difference in what is actually the carrier of energy in the cell, or again?
I see.
Everything is the same.
Yeah. So ATP is kind of in the mammalian cells, right? So we work in human systems or mouse systems. So for stem cells, we went with mouse because obviously human stem cell, that the amount of administration to look at that cell is enormous. So we went with mouse for that reason. So in mammalian cells, ATP is kind of the prevalent source of energy. So that's how you can deplete basically most of the processes. But it's true, there are, for example, other nucleosides, a lot like GTP, for example, right, is being used for actually nuclear transport across nuclear membrane, nuclear envelope. So this is something that we're actually looking into it. So it's, to my knowledge, it's not so widely spread as ATP, but you're right, there are other, I would say, minor sources that certain very specific processes tap into, but the majority of the cellular processes in the mammalian cells, in higher eukaryotes, would be ATP.
And the properties of the cell will not change if they were different. Again, so it's similar to my first question, because there are other animals that don't use ATP.
I see. Okay, so this is it's interesting, right? So if you would feed a cell that runs on ATP with GTP, it would not be able to use it because it has a specific receptors, right? Basically, the way how the ATP molecule is being used. So it's generally, right? Basically, these proteins that use these ATP or GTP have specific pockets where these actually very nicely sterically fit into, right? So it's given by their molecular geometry that they fit there. So where ATP works, GTP won't. But if you view it purely as a net energy source, then in theory, right, it's basically hydrolysis as hydrolysis. You break off a phosphate and you obtain energy.
Yeah.
So in that sense, it should be equal. Yeah.
Yep. Okay, one last thing. This is not a question, but I will make you host. So
Sure.
I will give you absolute power to terminate.
Yes.
In the meantime, I checked that you are right. Dejean did use the word soul.
Yes. I was so excited when we saw these phases, right? That of course I went to the source. I went to the Bible. So I looked how did Dejean do it? So that's because originally, right, it was liquid and solid, but
it is liquid and solid, so it's okay.
I have a simple question. When you revert a differentiated cell back to a stem cell,
yes.
Does it shrink and homogenize?
That is actually a very good question. I do not know, but it is possible to do. I have not done that experiment yet. And I have not seen images of these type of cells with basically these type of markers that I would be able to analyze with our techniques. But it's a very interesting thing because there is this technology of like induced pluripotent stem cells, right? So you can go basically from any differentiated cells, you can go backwards. That would be super cool to see actually if we can dissolve the heterochromatin, the gel patches, and make it homogenized. That's a very interesting idea. We can try it.
Go for it.
That's actually, well, you know, it's actually by now standardized protocol how you do these induced pluripotent stem cells. So I mean, it's, you know, it's not easy, but it's very much doable.
Very good. So other questions? Other suggestions what Alexandra should do rapidly? There are two suggestions. Do pluripotent cells and also study origin of life. Don't forget about this from the beginning of the talk.
I'll be busy for a while, yeah.
Other things. Well, if there are no other questions, then let us thank Alexandra once again. And perhaps let's stop recording.